Candida albicans is a microorganism, and it is a part of the fungal kingdom and belongs to a division or phylum known as Ascomycota. This division is very diverse and contains over 32,000 species. These species can vary in different ways from cell size to their reproduction process. Advancements in healthcare have caused an increase in fungi growth and therefore knowledge. This in turn results in more diagnosed infection cases. C. albicans has a eukaryotic cell type. It is a chemoheterotroph, meaning it requires organic compounds for energy and carbon. This is similar to animals, but they absorb their nutrients instead of ingesting them. They may be aerobic or facultatively anaerobic, but there are very few anaerobic fungi. Yeasts tend to be facultative anaerobes and molds tend to be aerobic. In most humans, it colonizes in mucous membranes, gastrointestinal tract, genitourinary tract, and the epidermis. It is a commensal organism, but can turn into an opportunistic pathogen. Normally, it does not cause problems in healthy individuals when kept in balance with other normal flora, but under certain conditions, it is able to undergo morphological changes and can cause problems for the host. It is widely distributed throughout the human population and is a low-level colonizer. Generally, it is harmless, and although it is harmless to normal individuals, it can be problematic in those with suppressed immune systems because of drug treatment or other immune disorders. Its ability to form biofilms on living and non-living organisms is a major reason for its high prevalence and pathogenicity. Because of this, it is able to form biofilms on medical equipment and is a major health concern for hospital-acquired infections. The phenotypic transitions C. albicans exhibits contribute to differences in cell structure and colony morphology. Pathogenic fungi tend to exhibit dimorphism. Because it is dimorphic, it is able to take on different forms at different temperatures, pH, and nutrition requirements. Its two forms of growth are a factor of its virulence because, as a yeast, it's able to attach to epithelial cells, and in its pseudohyphae form, it is able to further invade tissues. This increases its pathogenicity and makes it resistant to phagocytosis. Yeast-like growth tends to be at 25 degrees Celsius and at a pH of 5 below. Yeasts are unicellular and they reproduce by budding. This is an example of a yeast right here. Mold growth occurs at 37 degrees Celsius and at a pH range near 7, but usually below 7 and above 5. Molds are multicellular and they produce vegetative and aerial hyphae. This is an example of pseudohyphae, and this is an example of hyphae. These dimorphic abilities allow C. albicans to go from neither benefiting or harming its host to becoming a harmful pathogen. Fungi, whether a yeast or in hyphal form, make use of different reproductive strategies to produce offspring. Asexual spores produce buds known as blastoconidium. They do this via mitosis. These buds develop along pseudohyphae and divide unevenly. Chlamydoconidium are asexual fungal spores that form within a hypha. C. albicans is more virulent in its hyphal form. Here are the buds. Here is the pseudohyphae, and here's the chlamydospores. C. albicans has specific nutritional adaptations. Yeasts tend to grow at a more acidic pH than bacteria, around a pH of 5. C. albicans can grow in high sugar and salt concentrations where bacteria normally cannot grow or wouldn't be able to withstand the osmotic pressure. Its ability to grow where concentrations of salt or sugar are higher contributes to the infection cases in people with uncontrolled diabetes. It is also able to grow in low moisture regions where bacteria growth may be inhibited. In warm and moist regions, like the mouth and genital region, they are able to easily overcolonize and disrupt normal microbiota. This occurs when certain requirements are present. Exoenzymes like hydrolases allow for C. albicans to break down and take in nutrients from the environment and enhances its survival. They secrete these outside of the cell. This photo demonstrates Candida albicans colonies on a yeast extract peptone dextrose agar. Since C. albicans is a regular part of a human's normal flora, it does not normally infect healthy individuals. When infection does occur, it is able to infect many different parts of the body and may cause localized or more severe systemic infections. When infections occur, it is normally due to an opportunistic overgrowth when the competing microflora are suppressed by antibiotics or when some immunodeficiency is present. Candidiasis is a candida infection typically on the skin or mucous membranes. It is commonly the cause of vaginal yeast infections, oral thrush, and cutaneous candidiasis. Oral thrush tends to be one of the first clinical symptoms noticed in HIV-1 patients. Candidemia is a candida infection of the bloodstream. Candidemia can result from untreated candida infections, and some risk factors include immunosuppression, major surgery, and placement of medical devices such as a feeding tube or catheter. Symptoms can resemble those of bacterial sepsis. Candidemia infections lead to long hospital stays and increased mortality. Oral thrush. It is a normal part of the microflora of one's mouth, but causes infections when it overgrows. Symptoms of oral thrush are white spots in the mouth that may bleed when touched, a painful sensation in the mouth that may cause difficulty with eating or swallowing, redness inside the mouth or at the corners of the mouth, loss of taste, and a cotton-like feeling inside of mouth. Treatment consists of antifungal medications, usually in the form of a pill or liquid. Medications commonly used are nystatin or fluconazole, which interfere with fungal growth by inhibiting ergosterol. Ergosterol is a critical part of the fungal cell membrane. If left untreated, it can lead to systemic candida infections, which can be more severe. This is a picture of a healthy tongue, and this is a picture of a tongue that may have candidiasis. And this is a picture of a tongue that definitely has it. Notice the white spots and the redder regions. When candida invades the human body, it can infect internal organs like the liver, kidney, osseous tissue, and brain. Candidemia results from infections that became systemic. Symptoms include fever, kidney failure, and this may lead to shock. Diagnosis of candidemia is done by isolation from a blood sample. Treatments may include IV doses of fluconazole, caspofungin, mycofungin. The CDC estimates that the occurrence of candidemia infections in the United States is nearly 25,000 cases per year. Risk factors for this disease are typically dependent upon the individual because Candida is a commensal organism. Risk groups are people with weakened immunity, HIV patients, individuals on broad-spectrum antibiotics, or those with autoimmune disorders. Diabetics are also at risk, especially those with uncontrolled diabetes. IV drug users, and high mortality rate has been shown in smokers versus non-smokers with infection. Doctors may ask questions revolving around diet, recent usage of antibiotics, and medical history when trying to diagnose Candida infections. Physical examinations can be done in certain situations. A problem that arises when trying to treat infections is the increasing drug resistance due to antifungal medications. This may be because of antifungals as a primary treatment for fungal infections, not properly diagnosed. Also, since C. albicans is a part of a normal microbiome, it is impossible to completely treat an individual and rid them of this organism. It is only possible to maintain its growth and keep it from over-colonizing in order to prevent infection. Control and prevention may also be helped by improvements in healthcare delivery, and this can be done by keeping catheters clean, educating individuals on what a healthy microbiome is, and ways to maintain beneficial microflora